I'd like to first thank the organizers for preparing this nice meeting and also giving me the opportunity to present this morning. So this is third ACURE, and it's also my third attendance, and it's very nice to see the growing audience. The last ACURE in Barcelona, we presented the data of impact of LV unloading on left atrium, right ventricle, and other organs. So our lab has been interested in the non-LV effect of LV unloading. And it's fascinating that a lot of these effects can be explained by hemodynamics, but we thought there may be other mechanisms that may play important roles during the LV unloading for inter-chamber and inter-organ communications. This is important because when we look at the LV unloading as a therapy, we have to heal the whole body. So we need to look at other organs too. There can be a lot of mechanisms that play roles. It could be neuronal regulation, hormonal regulation. We got interested in the exosome-mediated regulation. So exosomes are very small particles that are released from cells, and they share the common membrane to the original cells. So they're very stable outside the cell, and they can diffuse, they can even travel in the bloodstream to the remote site, where they can be taken up by the recipient cells. And these exosomes contain different things. It can be protein, DNA, RNAs. We focused on microRNAs because they're very small, and they can be packaged in these small particles a lot. And microRNAs are thought to play important roles in the exosome-mediated intercellular regulation. MicroRNAs are short RNA that can bind to mRNAs, messenger RNAs, that are ready to translate proteins. The binding of microRNA to the mRNA inhibits protein translation or even degrade mRNAs that are going to translate proteins. So long story short, the donor cells can regulate the protein expression of remote cells using exosomes. So to look at the impact of unloading on exosomes, we designed a pig study. And this is a percutaneously induced myocardial infarction model. We worked on the pigs 1 week after MI because the reason we chose this time point is that in the very, very acute setting, a lot of cells dying from the MI are releasing exosome-like small vesicles, and we thought we were going to get a lot of noise from this data. So we wanted to focus on the impact of unloading. So we first drew blood. Pigs underwent LV unloading using Impella for 2 hours. And we drew another blood at 2 hours. In order to understand how the load impacts exosome profiles, we also included the animals that underwent LV overloading. LV overloading was induced by creating aortic regurgitation. So we made holes in the aortic valve by puncturing it. And after the aortic regurgitation, we were able to make a lot of regurgitation into the left ventricle. And we saw an increase in LV endoscopic pressure, indicating that the LV was successfully overloaded. To draw the blood samples, we put catheters, one in the coronary artery ostium and the other one in the coronary sinus. And we drew the blood simultaneously, so we were able to see the transcardiac gradient of exosomes in this setting. Here are the blood samples, and we isolate the exosomes, extracted microRNAs, and we sequenced these microRNAs. And in total, we found about 900 million microRNA reads, and these reads were mapped on the known pig genome database. And about 70% were mapped. So it's not perfect, but we were able to map about 2/3 of the sequence reads. And we compared these microRNAs before and after the coronary circulation. So we defined the ones that disappeared from the coronary artery blood to the coronary sinus blood as the ones that are taken up by the heart. And the ones that appear newly as the ones that were released from the heart. So we have coronary artery blood, coronary sinus blood, and in 3 pigs after unloading and 3 pigs after overloading. So this is the overall sample picture. And we first looked at the changes from baseline to unloading. So we had a lot of microRNA that appeared as the ones that appeared in the blood. But we thought many of these are just bystanders. So we looked at those that showed same trends in all 3 peaks in each group. And we found 39 that were uptaken and 7 released in all 3 peaks after unloading. Same way, we had 33 uptaken and 32 released after overloading. Now, we thought if these microRNAs are responding to the changing cardiac load, it should show the opposite changes to opposite change in the load. So we looked at the microRNAs that showed opposite changes after unloading and overloading. Using this approach, we found no microRNAs that were uptaken after unloading and released after overloading, but we found 6 microRNAs that were released after unloading and uptaken after overloading. Here's a list of microRNAs, and interestingly, not many of them were so very well characterized in the setting of cardiovascular diseases. And we are now looking into these microRNAs in our samples. In the first analysis, we did a before and after comparison, but we also looked at the samples only after, by direct comparison. So coronary artery blood from the unloading and overloading were compared, and so was coronary sinus blood. Using this approach, we can use the copy number as a factor, so the statistical power gets stronger, and here's the results. So this is called volcano plot, and the dot represent specific microRNA. So if there is a lot more microRNAs after unloading compared to overloading, it will be shown on the left side of the figure. And if it's more after overloading, it will be shown on the right side of the figure. The y-axis is the statistical power, and these bars show a p of 0.05 after the correction for multiple comparisons. So as you can see here, there's not so much microRNA that reached statistical significance in the coronary artery blood, indicating they're pretty similar. But in the coronary sinus, we found a lot of microRNA showing difference in unloading and overloading. So this data supports that there's a lot of active uptake and release in the heart during unloading and overloading. In summary, we found a lot of microRNAs that showed transcardiac gradient, and some of them show opposite changes to unloading and overloading. But this is a very early stage of our research, and we don't even know if this increased release is whether an active secretion or just an overflow from the cells that are producing a lot. We don't know which cells are producing these exosomes. We don't know where they go and what they do. So there's a lot of things we have to work on, and this will be our homework till next ACURE. Hopefully we can update you guys. But the bottom line is that exosomes do show different microRNA profiles in cardiac unloading and overloading, and they may play important roles during the unloading for cross-organ communications. Before ending, I want to thank all the people who worked on this project. Olympia especially worked on hard to get these data, but unfortunately she was not able to come this time. She and Kelly's work were essential for getting the in vivo data and collecting bloods. And people in Sasmita Safu's lab were experts in the exosome biology. They helped us a lot in extracting exosomes and analyzing our data. And to Roger, who also couldn't make it this morning, but I want to thank him for his support and also creating this nice environment in Mount Sinai to do this kind of collaborative research. Thank you very much for your attention.
This lecture is now open for questions. Kiyo, that's really elegant work. Realizing this is something you're just starting on, have you altered the timing yet of unloading? So thinking about the DTU study, have you looked at this prior to the infarct or prior to reperfusion?
So these data are only during 1 week after MI, but we also have a project that we are starting right now. So hopefully we can collect the blood samples in more acute setting. And probably sequencing is not a good idea because of noise we get from the dying cells, but we can always look at the microRNAs in the blood without like PCR and see the difference compared with tissue data.
We have time for one more question from the audience. Naveen?
Kiyo, that was a terrific talk. So, it's always challenging with microRNAs and circulating microRNAs versus tissue. You, at the end, very nicely illustrated all of the questions that are coming out. So, based on the panel that you showed, though, of the 6 microRNAs, I saw miR21 sitting there, and that's kind of a very popular one.
Yes.
Seems to have implications in a lot. So, just out of curiosity, what is the next step? I mean, do you think that, is there a way to do are you going to do more of an omics-based approach to look at screening for more microRNAs? Are you going to pick an integumer and go after miR21? Is the goal to try to replicate or enhance the effect? I'm just trying to understand sort of the direction you want to take this in.
I think we would try to look more detail into the data. So we are interested in the LV load. So we would take a look at the end-disk pressure and see the correlation between the copy numbers, and probably looked at the 3-5 top microRNAs, and then do PCRs on the tissue in the heart, kidney, other organs to see what is really differentially regulated during unloading. Then we'll design a study to specifically regulate these microRNAs.
It's a fascinating connection because what you're starting to see is that connection between hemodynamics and biology. And I think that's still a huge question that still remains unanswered, is how do these hemodynamic devices, how do they change the way cells respond in a pretty short amount of time, in 2 hours? So really fascinating.
Thank you.
Dan?
Just one quick question. I think one of your last conclusions was really provocative in that these microRNAs may provide a signal where one organ can communicate with the other. Is that, I think that was a really interesting message. But in your data slide, you showed that the levels of the mRNAs in the arterial blood, which would be the means of communication, right, was really not affected, and it was really this difference. So what are your thoughts about the hypothesis and that observation? And is it that what are your thoughts?
Right. That's something I thought too. If microRNAs in the coronary artery blood is the same, it's probably the microRNAs that were circulating or correcting in the lung and comes back as clean blood. So there may be something that are more uptake, and uptake of the heart may be more important. We really don't know what they're doing, and I think the key is to analyze the tissue and see if these microRNAs are actually up or down regulated, not only the blood. And then we'll have better clues, and hopefully we see the difference in the kidneys and liver, or major organs, and we can proceed. But unfortunately, we don't have the data yet.
I mean, it'd be interesting actually to hear from Dr. Kloner about this, because this is kind of like the idea of remote preconditioning. So those signals are not related to microRNAs, right? These are different,
well,
like
no,
heat
there
shock
actually
proteins.
are some studies in the literature suggesting that microRNAs might be one of the mechanisms for remote ischemic conditioning. It's known that some sort of humoral factor is likely involved, because you can condition a limb, let's say, take the venous blood, give that blood to another animal and reduce infarct size. So there's some humoral factor. Exactly what it is sort of has been elusive, but mRNA is one of the potential mechanisms.
Okay, so with that, we will continue for the second talk. Thank you very much, Kiyo.
Thank you.